So again, I'm delighted to be here to be able to present in this session. Thanks to the organizers, and also to the point that Soyon discussed. I think we've been going through some very difficult times with COVID, and then more recently with George Floyd in Minneapolis and Black Lives Matter, and all this racial tensions that are there. So it's really important for us to consider and think as a community about how we're going to deal with this. So again, I just want to emphasize that it's important to advocate for diversity in research, and to remove all barriers from research. And I think we should all be thinking about this as we move forward. So my interests are in chronic brain inflammation, and what I'd like to do is discuss some of the newer studies that have come from my group. But more recently, I would like to give you an insight into how I've come to this space. So before I came to the neurotrauma field specifically, I had worked in aging. And I was really, when I was setting up my lab several years ago, I was really interested in this interaction between acute brain injury and normal aging. And this figure here, which is a Perspectives article from Doug Smith and Willie Stewart from Nature Reviews Neurology, is very interesting. On the y-axis, we have this pathology score, and then on the x, we've got an age-related score. And the idea here is that in the absence of traumatic brain injury, pathologies such as amyloid, beta plaques, and inflammation accumulates as a normal function of age. And Doug and Willie Stewart were hypothesizing that perhaps after severe traumatic brain injury, as shown here in this red, you have this spike in pathology, okay, which occurs after a main traumatic brain injury, and oftentimes it resolves. But there is probably a proportion of individuals where this pathology does not resolve, this acute phase response does not resolve, and then it leads to this accelerated pathology and shifting of the clinical threshold, whether that be development of dementia or something else, to the left. So you start seeing this shift in turn with accelerated aging or dementia-like phenotypes. And if you model and consider mild traumatic brain injury or concussive-like brain injury, perhaps some repetitive mild traumatic brain injury hits would also occur. So again, you can sort of see this clinical threshold moving to the left. Interestingly enough, and I think very nicely done by James Cole and the Imperial group with David Sharp, have used neuroimaging modeling to look at predicted brain age in healthy individuals. And this is a very interesting study where they looked at structural MRI from 1,500 individuals, and then they predict what is the normal age of that scan. And then they applied that big data and MRI imaging analysis to traumatically brain injured patients and a healthy control population. And from these estimates, they determined that, TBI patients were estimated to be older with a mean predicted age of between their chronological age and their estimated brain age due to MRI of approximately 4.66 years when it comes to the gray matter, and almost 6 years when it comes to the white matter. So this predictive model would suggest that this may lead to predictive cognitive impairments and correlates very strongly with time post-injury, suggesting that brain tissue loss increases throughout the post-injury phase. So what are the mechanisms that may be involved in this? So one of the questions that we had asked, and many people are focusing on amyloid and tau, et cetera, but the immune system and innate immune dysfunction plays a significant role. So one of the questions we had were, would immune senescence or dysfunctional microglia play a role in shifting this curve, and would that be something of value to investigate? So let's think about chronic inflammation more broadly. So the brain is a highly immunogenic organ. So the primary innate immune cells are the microglia, and the sister cells, the astrocytes, and they regulate normal tissue homeostasis with neurons. But as the brain ages, there's this natural phenomenon where it's called inflammaging, where there's a loss of brain tissue volume, and it's also a change in functional compartment or functionality of these glial cells, and particularly the microglia, where they develop this senescence-associated secretory phenotype, this senescence phenotype, and that results in a priming of the microglia. And then after injury or chronic neurodegeneration or disease, you have this heightened inflammatory response, this neuroinflammatory response, where you have these loss of these protective factors, which may include clearing of damaged proteins by phagocytosis and all these beneficial effects. And you have the more inflammatory microglial response. And then this leads to this feed-forward loop where these damaged microglia are feeding into these damaged neurons and resulting in this dysregulated and reactive phenotype. So Adele has presented this data before, but this is some of the very important early evidence to suggest that inflammation, chronic inflammation is seen in humans after TBI. And these studies, as Adele mentioned, were performed by Dave Sharp's group in Imperial and demonstrates at months post-injury, up to 17 years post-injury, the binding here is for the TSPO ligand PK1195 at the level of the thalamus. And most, compared to age-matched controls, you can see there's a very high expression of these inflammatory mediators at that time point. Oh dear, I think I've lost my... Hello? Hello? Okay.
Everything all right?
It just broke down for me. I think I'm back up and running.
Yeah,
Hello?
you're back up and running.
Okay.
All the
Sorry
slides are
about
good.
that. Yeah, thank you. So just to confirm those studies, when you look at pathological studies, these again from Willie Stewart's group at Glasgow, have confirmed in approximately 25% of those individuals who go to autopsy and have actually survived beyond 1 year post-injury, there's a highly inflammatory reactive white matter, which includes these MHC class 2 or CR343 reactive microglia associated with thinning of the myelin and some reactive engulfment of actually myelin basic protein. So again, further evidence to suggest even after a single severe traumatic brain injury that there's a chronic persistent inflammatory response. So this is the clinical evidence that we have been using to explore and model in our rodent systems. And what we use is an experimental brain injury model called controlled cortical impact, where we induce a local focal contusion injury on the left parietal cortex of the injured mouse. And then we can look at the development of inflammation and particularly microglial reactivity over time. So what these slides are here are some two-photon imaging of microglia which overexpress the CXCR1 GFP. So each individual cell here is the microglia in the normal non-injured brain. And what you'll notice is that from these IMARIS reconstruction analysis, the shape of the microglia, they're small cell bodies and very many processes. But after a week post-injury, there's a dramatic change in the morphology of these microglia, such that they have fewer projections and they have a large cell body, and perhaps these processes are thickened as well. So this is a very useful way to quantitatively assess the activation profile of microglia. And when we do this in cohorts of animals up to 1 year post-injury and quantify the number of surveillant ramified microglia versus these hyperreactive microglia, what you'll notice is that there's a loss of surveillance microglia that is extended throughout the whole time course up to 1 year post-injury, and a greater number of these highly reactive microglia. The peak occurs at 1 week post-injury, but even at 1 year post-injury, it's evident that there's still a high number of these reactive microglia present in the injured brain compared to age-matched sham controls. You can then use flow cytometry to actually take those microglia out of the injured brain of these rodents, and then use do some functional analysis. So this experiment here is a flow cytometry plot of microglia which are labeled by CD11b, CD45 intermediate population. And one of the first things to note here is that even at 3 months post-injury or 90 days post-injury, there's no there's no infiltrating immune cells, neutrophils or monocytes who are trafficking to the brain. So the primary innate response is driven by the microglia, and there's increased numbers. So there's increased number of microglial counts, and then these scatter profiles suggest that they're altered in shape. But if you go and probe for their function within these cells, it's evident as well within these chronically brain-injured microglia that they have upregulated TNF and IL-1, two very typical pro-inflammatory cytokines. They also upregulate senescence, so BCL2 and PH2AX. And when it comes to some of their functional capabilities, they have impaired functional capacity for performing phagocytosis. So essentially, if you offer a bead which is labeled with a FiTBI or an amyloid protein which is labeled with FiTBI, we'll notice that the chronically injured animals have less ability to take up that cargo and perform that critical functional aspect. Another thing that was of interest to us when we were scanning the literature was an enzyme system called NADPH oxidase. And what we have been able to show in these pathological studies is that this enzyme NOX2 or NADPH oxidase is chronically expressed. These are examples at 1 year post-injury within the injured cortex and the injured thalamus. And what you'll notice, hopefully you can see here, is that there's NOX2 expression within these subregions, which co-localizes with some of these microglial-associated markers and a reactive phagocytic marker, CD68. And I think it's most evident here at these far distant sites from the injury. So it's not at the local contusion site, but at distant sites such as the hippocampus, or sorry, such as the thalamus. And this chronic reactivity of NOX is also coincident with oxidative damage, progressive neurodegeneration, as we've demonstrated in these quantitative analysis, white matter damage, and persistent cognitive deficits when we use these several tests for cognitive functions such as the Mars-Water maze. So why is NOX important? Well, you know, there's a lot of evidence from experimental and preclinical models that NOX is reactive and is stimulated by various pathogens and damage-associated patterns such as interleukins, amyloid or misfolded proteins, inflammagens, and toxins. And when that occurs, it produces free radicals such as superoxide and drives NF-kappa B dependent signaling, which then increases ROS and amplifies this pro-inflammatory response, which can then increase and lead to neuronal damage. And that local neuronal injury actually results in this feedback mechanism, which is this chronic reactive microgliosis. So we and others actually have been very interested in this pathway and have demonstrated that indeed the repetitive mild or moderate to severe level traumatic brain injury is a very strong stimulus of this NOX-dependent pathway in microglia. And if you actually, and it progresses, and if you inhibit that, either using genetic approaches or pharmacological approaches, you can actually improve or suppress the inflammatory phenotype and improve the neurological recovery within those animals. So just to summarize this kind of introduction part of the study, I think there's significant evidence now in humans to suggest that, at least at the severe injury, at the level of severe injury, that TBI produces chronic reactivity of microglia, particularly in white matter regions. It's been shown quite well that, you know, chronically activated microglia are pro-inflammatory and senescent and have impaired ability for key functional responses such as phagocytosis. And we and others have been able to show that this protein NOX2 is really quite important in driving this pro-inflammatory microglial phenotype. It is chronically expressed and it's an important therapeutic target. And I don't have time to show you the data today, but actually if you regulate it and inhibit it in a very precise manner, you can actually, more than just reduce the pro-inflammatory phenotype, you can boost the anti-inflammatory type of mechanisms and boost trophic factor production from these cells. So a very important target from experimental point of view. So what I really wanted to present quickly was some new work that we've done and used. We've taken advantage of these new class of drugs. These are called colony-stimulating factor 1 receptor inhibitors. And essentially, these were developed by Kim Green's work at UC Irvine, where he's developed, in collaboration with a drug company, first and second generation inhibitors. Now, the key thing about these inhibitors is that they CSF1 receptor signaling is required for neuronal survival in the adult brain. So if you inhibit this signaling, you can deplete microglia from the injured brain. Now that's shown here in these analyses, where you see each individual plot here is a single microglial cell, and these are under the control adult mouse conditions. Then when Kim Green's group administered PLX3397 in the chow, within 21 days, you can see that there's almost complete removal of these microglia. There's solely only 5% remaining. And actually, under those conditions, animals perform well. They don't have deficits in cognitive function, in motor function, all these other things. So it's a really unique finding. But more than that, if you remove the inhibitor, you can allow the microglia repopulate from these remaining cells. So there's this resistant population to CSF1 inhibition, and when you remove that inhibitor, the microglia can retile the brain, which is a really interesting idea and a really interesting tool for proof of concept research. And this is being used in Alzheimer's disease models, in CNS injury, and other ideas. And we wanted to use this to test a hypothesis that could we target microglia during the chronic phase of injury to see, are they really critical for these chronic neurodegenerative response that's occurring within these experimental systems. So the question was, can we remove chronic neurotoxic microglia after traumatic brain injury and then repopulate them with an anti-inflammatory or neuroprotective microglial population? And will this lead to improved motor and cognitive function recovery? So Rebecca Henry is a talented postdoc who's in my lab at University of Maryland where these studies occurred, and she took this on. So the idea here was it's a delayed microglial depletion model where we induce a traumatic brain injury or sham injury, and then at 4 weeks post-injury, we remove the microglia by adding this PLX5622. And this schematic tries to show you what's idea is happening. There's these, under non-injury conditions, they're the ramified cells, they become activated. Then you add the drug and actually you remove these activated cells, and then after a week you remove the inhibitor and then you repopulate them. And the idea being that these repopulated cells may be protective in some manner. And then Rebecca allowed these animals to continue to progress through 7 and 12 weeks post-injury, perform a molecular flow cytometry analysis and neuropathological analysis, as well as motor and cognitive function recovery tests. So the first thing to show is, indeed, this drug works the way it's supposed to work. So this is the control cells. These are P2Y12 positive microglia, and you can see they tile the brain very well. 7 days on PLX 5622, you can remove, 95%, like Kim Green's work has shown. More than that, flow cytometry analysis confirms this. So you have control vehicle levels, the PLX depleted levels, but then you remove the inhibitor, and you can see that these microglia can repopulate towards those control levels. So our model is in place, which was always encouraging when you set about these studies. So to show you some of the motor function tests and analysis that was done, the animals had a traumatic brain injury, and it occurred, and we tested for weeks post-injury, up to 4 weeks post-injury. You can see the vehicle treated, which are shown in red, and the PLX treated and shown in blue, had the same level of motor dysfunction on a motor coordination task called the beam walk task. Then at this time point, we randomized animals to receive the PLX chow or the vehicle chow. So that meant that under this time peak between 4 and 5, the microglia were depleted or not under the vehicle conditions, and then repopulated from week 5 all the way through the end. And what you'll notice, and I think is striking, is that there's a persistent impairment in motor function in the vehicle-treated animals. But those animals that had the repopulated microglia start to show improved performance in this motor task. And to confirm this, we performed a rotorad task as well at this very terminal endpoint at 12 weeks post-injury. And I think what you'll see here is these very striking trends to showing that there's an impairment in the ability of these mice to perform this motor task in the vehicle-treated groups. When you look at the repopulated cells, they certainly, those animals seem to do a lot better on this motor task, suggesting that there's improved motor performance. We also then moved to a more classical cognitive function task, and this is one of the tasks that we tested. So this is the Morris water maze, where the animals are in a submerged pool with a submerged platform, and they use spatial cues to try and identify where that submerged platform is. And what we noticed in the acquisition learning phase, that there was certainly a difference between sham injured and TBI animals, and perhaps there was an improvement or a slight trend towards improvement with those PLX treated animals. In a probe trial, which occurs on day 5 in this form, where the animals are trying to find the platform, but it's missing in this circumstance, we looked at the time in this escape quadrant. You can see that those vehicle treated animals had impaired performance, so they this time they spent in the escape quadrant is dramatically reduced. And in contrast, the PLX-treated animals had improved performance. And when you perform some additional type of search strategy analysis, I think the critical point here is if you look at the ability, the difference between sham and injured animals, the most highly injured animals use this random looping strategy to find the escape from this pool. And what's important here is that those PLX-treated animals, the ones that had microglial repopulation, use more of these systematic and spatial learning analyses. And this very much mimics like what's seen in the sham injury condition. So again, improved performance with this very delayed treatment paradigm. When we looked at lesion size, and we can quantify a lesion using various stereological methods, and essentially what you're comparing here is the size of the cortical lesion in the ipsilateral cortex, and this is the vehicle-treated conditions compared to your PLX-treated. There's a significant attenuation, and this was also seen at the level of the dentate gyrus, okay? So again, at hippocampal sites, there's preserved, there's reduced TBI induced neuronal loss. And when you looked at microglial and morphological transitions and the characteristics that I've showed earlier in the presentation, you can see that there's reduced numbers of these ramified cells, these normal cells, and there's increased number of these hypertrophic bushy cells. And that is indeed reversed or brought back towards control levels when you repopulate the microglia, again indicating that this repopulation, these cells return with an altered phenotype. Some transcriptional analysis which can be performed, and in this circumstance, this is a tissue level analysis to look at 770 genes related to inflammation and neurodegeneration. And I'll skip the component analysis, but I suppose just mirroring what Soyeon's studies have been identifying, if you compare the vehicle-treated TBI group versus the PLX-treated TBI group, some of those important pathways that Soyeon was talking about, the complement system, C3, C1QA, CYBB, TREM2, CD68, all these things are up in the chronic TBI group, but are significantly reduced in the PLX-treated group. So again, suggesting that we're having dramatic change on the inflammatory phenotype within that region, and oxidative damage is also decreased. And to bring it back to those prior studies that I introduced earlier on, this NOx-associated inflammation markers are also decreased. So NOx messenger RNA, but more importantly, NOx protein. And what we're showing here is NOx-positive microglia. So they are NOx2 positive, CD68 positive, IBA1 positive microglia. And what you can see is under the PLX-treated conditions, there's reduced NOx2 CD68 within the injured brain. Now, another group had previously shown that the inflammasome was actually downstream of NADPH oxidase. And the inflammasome, NLRP3 inflammasome, very important complex when it comes to age-related neurodegeneration. Alzheimer's disease models and Parkinson's disease models have shown that it's played a very important role in pathology and cognitive impairments. And it's a component system which includes an active inflammasome, ASC protein, and pro-caspase-1. And when it complexes, it can become active and produce this active caspase-1, which can then cleave the pro-IL-1 or IL-18 to form these mature cytokines, which then have inflammatory capabilities. And what has been shown in prior work is that TBI induces the NLRP3 inflammasome. So you can see whether it be NLRP3, ASC, or cleave caspase-1, they're all upregulated. But under the conditions of deficiency of NOx2 or NADPH oxidase, you have a significant reduction in these in the inflammasome activation. So again, suggesting that NOx2 is upstream of NLRP3 inflammasome within the context of TBI. So we wanted to explore that in our tissue sets, and indeed what we did see that there was chronic expression of NLRP3 inflammasome, and when it looked at gene expression profiles, that was reduced with chronic PLX treatment. And more so, if we pulled out those microglial cells from those injured brains, and we used a flow cytometry analysis and a FLICA cleave caspase-1 activity assay, what we'd shown is that indeed, chronically, microglia express more caspase-1, and this is certainly reduced under the conditions of the microglial repopulation study. And microglia also produce more active IL-1, and again, this is that important inflammatory protein which is driving a lot of the pathological events. So again, caspase-1, IL-1 these levels are reduced with PLX treatment. It is important to note, because others have studied it, that in the context and these changes that occur within inflammation and these glial signaling, that there was no effect on the astrocyte response. So these analyses, which are doing morphological assessments of GFAP positive astrocytes, show that under the conditions of TBI, there was no real difference between those two things. So just as we published this paper several months ago, there was another study which came out in Cell, which actually went several steps further, actually, which I was delighted to see. And they used several models. They used the PLX 5622 model to look at microglial depletion, but also genetic models. And what they demonstrated was that if you remove microglia during the acute injury phase and leave them absent from the system, you cannot improve the cognitive recovery in those animals. And what they showed was it was critical that you had repopulating microglia. So after the injury, they are removed from the system, but it requires this repopulation to actually stimulate neurogenesis and improve learning and memory using various tasks. And they went on to show that there was a very significant mechanism in play, and that is that the repopulated microglia were inducing IL-6 signaling in neurons, and then they were doing this, they were mediating this neuroprotection effect by this IL-6 trans signaling through the GP130 activation. So again, some further ideas that indeed it's not only the removal of these neurotoxic microglia, but the repopulation, which is critical for improved cognitive and motor function recovery. So just to summarize where the studies that I've presented to you today, under the conditions of chronic brain injury, when you remove the chronic phase microglial response, the normal responses in chronic, in wild type or vehicle treated animals, there's these chronic impairments in motor and cognitive function, reactive microglial phenotype, including NOx and NLRP3 inflammasome, and this leads to progressive neurodegeneration, hippocampal neurodegeneration. When you remove microglia during the chronic phase, and we did this by this PLX 5622, this so-called reset of microglia, you can improve motor and cognitive function recovery so that they have reduced swim speeds and times when they're trying to locate that hidden platform. They also have reduced inflammatory NLRP3 inflammasome and NOx2 activity, and this is associated with improved neuronal survival within the hippocampus and reduced lesion size. So I suppose some of these take-home messages and what we can offer as preclinical scientists is that I think much to what Adele was speaking to, is that the inflammatory response is a very chronic and progressive response, and perhaps the therapeutic window for treating TBI, particularly in the severe injury side, is much longer than we traditionally believed if we target microglia and evolving inflammation in a more regulated and precise manner. Additional things that we have been working on for some time is that NOx2 is an important regulator of this chronic phase response, and we've got some other data to support this. And I suppose one of the ideas here is that this so-called reset of microglia using these CSF1 inhibitors may be useful therapeutic agents to try and re-challenge, to remove the neurotoxic, this destructive phenotype, and promote this recovery and neuroprotective phenotype. Now, I will add that these are very preliminary preclinical studies, and as we know, we need to progress much beyond for translational research. But it is a useful proof of concept analysis to suggest that these type of approaches could be used to try and improve outcome in certain chronically injured individuals. So just to highlight the team who have played a major role in this, and these studies were conducted at University of Maryland. These studies were really led by Rebecca Henry, who is a postdoc, and also Rodney Ritzel, who's done a lot of the development when it comes to flow cytometry, which was a really advanced or functional analysis. I've more recently returned to Dublin, to Trinity College, so I'm starting a new program here in Dublin. And indeed, we have a lot of collaborators, including a long-term collaborator and former mentor, Alan Faden, and collaborators at UMBC, Greg Setzo and Willie Stewart in Glasgow. And just before I finish, I would just like to highlight one last slide, because during COVID, me, along with some other researchers in the United States, have been participating and organizing an additional webinar series that has been useful for the preclinical community in particular. It's called Current Topics in TBI. It's an online speaker series. It occurs using Zoom as the platform. It occurs every Monday, Wednesday, and Friday. It's very short. It occurs at 8 p.m. GMT. And actually, tomorrow we have coming up Umesh Venkatsan, who is a PhD researcher working at Moss Rehabilitation Research Institute, and he'll be discussing some of his research about systems neuroscience and human TBI. So just to plug that, and others who are excited about the current online Frontiers conference, you know, if you want to do some more of this, there are other avenues, so please check that out. Okay, so I will leave it there.
Okay, thank you very much, David. That was brilliant. Do you have time for some questions? I mean, I know we're massively over, but
yeah,
it is lunchtime. Do
yeah,
you want to take some 5 minutes for questions, and then you can always engage with people? I can leave this running, and people can keep carrying on asking.
Yeah.
So
let me try and
you can see coming up. Just while David is looking, just to remind you all that at 1 p.m. we have biomarkers starting up in session A, and then at 1:35 concurrently we have the second lot of the rapid fire presentations. These are early career researchers who have done a lot of really interesting work. Come and vote for your favorite. So over to you, David, for your questions.
Okay, so there's a few technical things, so perhaps maybe I'll get those out of the way first. So could you repeat how they repopulate microglia? So essentially, this is a new drug, as I mentioned, which is given to animals in their chow. So essentially, this is this CSF1 inhibitor, which is administered in the food. And this has been very well characterized. So if you need these details, please check out Kim Green's work. It's administered in chow, and the animals eat this up, and the microglia get removed from the brain. I would say that it is generally quite specific. There is evidence to suggest it's predominantly the microglia that are targeted, but there is evidence to suggest that there may be other macrophages that may have be impacted in that. Now, we've done a lot of controlled experiments to show that it's predominantly microglia responses, but that's another caveat of this experimental paradigm. So essentially how you do is you administer the food in feed, and then you can remove that feed, and then from that the remaining microglia, I suppose, that were that didn't get inhibited, they have this repopulation. Why did we choose 4 to 5 weeks post-injury? Well, that really was just a chronic time point within the experimental model system, where we think that you know, the most reactive microglia are progressing. They're very highly reactive when it comes to NOx activity, to inflammasome expression. So again, we didn't want to deplete microglia during the acute phase, where we believe they play a really critical role in trying to respond to this acute phase injury response. We wanted to focus on these chronic neurodegenerative phenotypes that are associated with chronic responses. So it was for this time that we chose that time point. Okay, Dave Sharp's question about resetting microglia and degeneration. And yes, indeed. So the question here is, does resetting this delayed removal of microglia and resetting of the microglia, that results in a related reduction in the long-term neurodegeneration when you quantify hippocampal neurons and the cortical lesions? So yes, it does seem to arrest the level of neurodegeneration. I wouldn't say it may add to a reparative type of environment that may lead, and we need to investigate this into the future, it may lead to more trophic responses and neurogenesis type of functional rewiring. But at the level of quantitative analysis, we arrest the neurodegeneration that occurs. And that is quite a useful thing, we believe. And that is as associated with motor and cognitive function recovery. Let me see. Since there is a difficult translation element from animal to humans, what do you suspect would be the best time to target these chronic inflammatory markers in humans? Yeah, no, this is really important, and I suppose it comes to what the work that Adele has been describing, and the requirement and need for really useful biomarkers of injury progression and during the acute phase and the chronic phase. So, it is a really tough question to address. How do these timings translate from rodent to a human? But I think it's really important to do a lot of work on the biomarker side. So we may have predictive biomarkers, and there's a lot of works that Adele mentioned, but also by Amy Wagner's group and many of the fluid biomarkers. And I think we're going to have a really wonderful session later on about biomarkers. So maybe some of those may predict when we could intervene for this type of therapy. So we're a little bit early with our biomarker research to identify the exact time point. But I think as we learn more about the biomarkers and how they relate to the injury progression, we will then be able to identify the window of opportunity to target these. I would say, one of the things and the advantages of preclinical modeling is that we do have this injury time point that it sets off this injury reaction, and then we can actually do in parallel, and we often do, although I haven't presented this data, take the serum and CSF and whatever else from animals, so we can relate the mechanisms of action that are occurring in these TBI animals and relate to how that to the biomarkers that are occurring within the blood or the CSF. And these are important goals of ours, to have a translational biomarker that will move from the preclinical model all the way to the human condition. Okay, I think. Yes, I just want to address again this peripheral myeloid population. So, as I said, most of these tissue-resident myeloid cells were not impacted by this PLX treatment paradigm, although we did identify skin myeloid cells, or CD45 positive, CD115 positive cells that may be impacted. So there's just a few unique populations. We're not sure how relevant they are to the injury progression and response, but in general, the PLX is a useful tool, and I will stress this is a useful tool to look at proof of principle studies. I think what's a better way of doing that is combining the PLX with the genetic models, where you can specifically target them in a very fine, useful way using Cree models, and I think that's a useful way to look at mechanisms of actions to avoid off-target effects due to PLX CSF1 inhibitor targets. But we're aware and cognizant of these effects. Okay, and then this last one. Yes, so the last thing, James Loane's question here. Did we see any effects of PLX on astrocyte response to trauma? So at the level that we have evaluated this, we have not. Okay, so I've shown some analysis of GFAP, reactive astrocytes in the chronic phase, and there was no change between the PLX depleted microglia and the non-depleted microglia with regards to the astrocyte response. Astrocytes certainly communicate readily with microglia through various mechanisms and cytokines and signaling, and that's really important for injury response and neurodegeneration. We'll add that, there's some work from John Godbout's group at Ohio State University who have done something very similar to this, except for removed microglia in the acute phase post-injury. And what they noticed was when you don't have microglia in the system, but astrocytes are clearly there, what they have shown is that there's changes in the astrocyte reactivity, their morphology, but also their transcriptional expression. So they are less inflammatory and all these other things. So this communication between astrocytes and microglia is really important to injury outcome. In the chronic phase, we didn't see any evidence, but I think at the level that we're looking at this, we probably need to do a deeper dive to explore this more carefully. Okay, so I think I will leave it there.
Perfect. Thank you so much, David, for sharing, and thank you for to all three of our speakers for a great session. Yeah. Obviously, we are going to be coming back at 1 o'clock for biomarkers, and then 1:35 for the rapid fire sessions. Great. Have a good lunch break, everyone, and hope to see you at the rest of the conference. Take care, David. Great job.
Thank you. All right. Bye-bye.